All right, so good morning, and thank you for letting me teach in this way. Although I'm actually surprised that we're not in a recess given the situation, but yeah, the UPR works in very funny ways. So, um, so what we're going to do today is complete our discussion about iron transport via the transfer and endocytosis process. And what I want to do is focus on this slide as a starting point, as a review of what we had discussed in our previous lecture. So the objective of this particular figure is to give us an overview of what is understood about metal transport by this particular protein. And so, as I had mentioned previously, this protein requires metal binding in order for it to be recognized by its receptor. And that particular interaction of the metal-bound protein with receptor is what triggers the process of endocytosis. And so, as we discuss a few of the details of this process, once the endosome is formed and enters into the intracellular environment, there is a decrease in the pH of that endosome from 7.4, which is the pH of the extracellular space of serum, and now becomes a pH of about 5.5. And so that acidification event plus potential chelation can lead to metal release from the protein. And so what I have here is sort of an overview of metals in general, not just of iron itself. But in the iron case, it's generally understood that iron has to get reduced to its iron 2 form in order for it to be released through the divalent metal transporter. Now, when it comes to other metals, this process of release from the endosome is not as clear, although it's speculated that if a metal is divalent, it would go through the divalent metal transport, just like the iron 2, and other metals possibly go through a chelation method, sort of a chelator that serves as a function of an ionoform to remove the metal from the endosome. So focusing on specifically iron in the cellular environment. So once you already have transported that metal into the intracellular environment, what happens next? And so I've alluded a bit to this idea of the iron trafficking. That's the formality of what happens to that iron as it gets distributed for use. So you're going to have some amount of that iron. So this particular picture is a little bit small, but it's just to give us this impression of where iron is going to locate. So you're going to have a portion of that iron that's going to get stored in a protein called ferritin. Okay, and so that protein is actually really important as our main mechanism for retaining iron. And so we're going to talk about that in a moment. And then you have a lot of that iron that is going to get functionalized. And so you're going to see movement of the iron into small molecules or into proteins that depend on it in order to be functional. So as I mentioned before, there is a transient labile iron pool, which is dominantly iron 2 based, although it is a mixture of iron 2 and iron 3, but mainly iron 2. And so that's constantly getting replenished. And so you're going to have that that transient but still consistent pool that exists that helps to supply the iron that's going to be stored, the iron that's going to be utilized. All right. So where the actual iron that's actually going to be utilized. And so we've talked about this idea that a large portion of that iron ends up in our actual blood cells, our red blood cells. And so the first thing is that the iron that is now reduced to iron 2 is going to get shuttled into the mitochondria. And within the mitochondria, it actually gets placed into a molecule called protoporphyrin 9. And so that is the molecule that once it binds a metal is what we commonly refer to as the heme group. So this small molecule is actually recycled in many different proteins. In the context of hemoglobin, as we know it, it serves as the iron transport function. And so that's where 75% of that iron that's ingested into the body is where it ultimately ends up. So another chunk of that metal, it gets exported to different binding sites. For instance, you have the formation of iron sulfide clusters. And so here are some of the classical forms in which these clusters can form. You have the what is referred to as the 2-iron 2-sulfide site, which is not necessarily a cluster, but is one of the modalities in which this iron sulfide can actually form. And so as you can see with all of these, these particular iron sulfur clusters, you're going to see an affiliation of cysteine groups to satisfy the coordination of the iron itself. And so just to briefly talk about the coordination mode, you have these irons that are in a tetrahedral coordination, where you typically have at least 2 or more sulfide contacts with the iron, and you have cysteine through the thiol groups, also through sulfides, that are satisfying the coordination of that iron. And so you have here a 3-iron 4 sulfide cluster, and then the classical 4-iron 4 sulfide cluster, which is this cubane type structure. And so the significance of these type of groups is that they partake in important roles of electron transport in proteins, and can, as a consequence of that, can also be very important for redox regulation enzymes. All right, we also have a significant amount of that iron getting trafficked into that protein ferritin. And so this is the iron storage protein. You have this cartoon representation of this protein as it surrounds this basically really big core of iron. And so let's talk about some of the features of this particular protein. So it is found in plants, animals, and bacteria, serving this very same function as we see it in humans. It is a 24 polypeptides, consists of 24 polypeptide subunits. It's pretty big, 480 kilodalton. And so it's called a nanoreactor core because essentially what it's doing, it's forming iron nanoparticles, iron oxide nanoparticles. And so the way it's facilitating this is that it actually consists of some of its subunits of a ferroxidase functionality. So what happens is that you have iron 2 getting shuttled into the protein's core, and then this ferroxidase oxidizing the iron from the iron 2 to iron 3 form. And as we discussed in our previous lecture, iron 3 is quite insoluble. So essentially that oxidation event is an important event because it encourages the formation of these nanoparticles of iron so that it precipitates. So that is the goal of here. It's an intentional precipitation of the iron in order to retain it within the protein's core. And so effectively, what this protein is doing is getting a significant amount of iron inside of itself so that you essentially have a large amount of iron within the cellular environment. So technically speaking, this is iron that is insoluble, but it's regulated in such a way that when there is a decrease in the iron below the homeostatic level, this iron will get functionalized to become soluble. So you're basically raising the concentration of iron that's accessible within the cellular environment to about a millimolar amount. Keep in mind that that's actually not soluble iron. It's iron that can become solubilized. Because remember, the soluble iron that is present within the cellular environment, it's at the micromolar level. So as such, the deletion of the gene that expresses the ferritin protein is actually a deadly event, right? So we cannot function without the presence of this particular protein, as we then wouldn't have a mechanism for storing our iron. All right, so with that in mind, I want to return to this figure that we looked at our very first lecture, when we were discussing this idea of the maintaining a homeostatic control of metals. And so here we have this perspective of essential elements and their dosage and a response. And so you have that sweet spot that triggers, the optimal amount of that particular metal. But then you have, if you go too far below, you have a deficiency that, especially when you're too far below, can have an impact on survival. Whereas on the other end, if you go too high, you can lead into toxicity and then ultimately into a lethal amount in the body. So let's talk a bit about how the body can try to regulate the homeostatic control of this metal. Because as I mentioned in our previous lecture, we don't have an effective molecular mechanism for release of iron. So we have to have a pretty controlled amount of iron that we intake into our bodies. But we do have this mechanism imposed in order to control when we're slightly below or slightly above this homeostatic level. And so when the iron level in our body does fall below the optimal level, what our response is, our cells will mobilize the ferritin in order to access that stored iron. So let's think about this idea of mobilization, because as you have seen in biochemistry, the body has its mechanisms for mobilization. So you have seen this in previously, where the body can mobilize proteins, it can mobilize polysaccharides, triacylglycerol, nucleic acids in order to break them down from their polymeric form into their monomeric form, and then into their monomeric form, break them down even further into their corresponding metabolites. And so we have seen the role of different enzymes in order to facilitate this. Proteases enabling the formation of amino acids, amylase enabling the formation of monosaccharides, lipase for the production of fatty acids, and DNase, RNase for the production of nucleotides. And then we know further, the breakdown of the amino acids, the monosaccharides, and the fatty acids can lead to the formation of metabolites that will partake in different biomolecular cycles, primary one being the citric acid cycle in the mitochondrion, which can produce 90% of the energy that's used by aerobic human cells. Whereas nucleotides, the catabolism of these particular monomeric species are not as important in terms of the amount of metabolic energy that can be yielded from them. All right, so with that in mind, let's think about how ferritin might be mobilized in order for us to be able to gain access to that iron. And so it has been proposed that perhaps there is a digestion of the ferritin by lysosome. And so, excuse me. So within this idea, what essentially would happen is that you would have that protein become proteolyzed. So proteolyzed means there's some sort of enzyme that is specific to the protein to be able to cleave it. And this typically is enabled by acid hydrolysis. The problem with this model is that when you have an acidification event, we know that that can help to solubilize the metal, but a sudden flux of acidification, and especially as it can enter right into the cytosol itself, can be quite toxic. And so it's not likely that this is the actual route by which ferritin is going to be mobilized in order to release its iron. More likely is that you have this sort of controlled, this regulated unfolding and opening of the ferritin pores to enable this release of the iron, this more controlled release of the iron. So it's proposed that there's a signal sent indicating that the iron resources are becoming depleted. And so that there is a need for iron to be delivered into the cell environment so that that iron can be released from cells. And so the idea is that that signal reaches the ferritin, and that results in an opening of the pores that's now going to lead to an event such as the presence of reducing agents to reduce that iron 3 to the iron 2 form. And so that reduction event is actually an important one, as it can start to enable that iron to become mobilized. Remember, iron 2 is more soluble, and it's aided by the presence of chelators that will help to coordinate the metal and further facilitate the solubilization and also the release of the iron from ferritin itself. So this seems to be the more likely event, a controlled signaling and also reduction coupled with chelation to be able to facilitate mobilization of the iron within the core of the ferritin protein. All right, so let's look at the other end of this process. When we're just above the optimal level, now we're starting to get an overdose of iron in the body. So there are some methods with that the body can respond, such as intracellular chelation to try to control that iron and make it not available. You also have processes known as extrusion. So what that means is that you have a transporter that's activated to expel the excess metal from the cell. And so a metal will then be shuttled to a storage compartment, also to make it inert. So these are ways that you can control that iron so that you don't have this excess labile iron that can potentially lead to an excess amount of reactive oxygen species formed, given that that can lead to, as we've already talked about before, a certain amount of toxicity. And then you also have the process of exclusion, and that means that the metal's route of entry is removed or inhibited. And so you can imagine that you would have maybe a suppression of the transferrin receptor to prevent an increase of iron-bound transferrin from entering to the cells. So these are mechanisms that are in place for dealing with an excess, but again, there will be a slight excess of iron above that optimal range. But when we're talking about really large excess, the body has a hard time dealing with that, as we don't have a very effective physiological route for clearing that iron. And so there now we're starting to talk about more extensive approaches, medical approaches, and be able to deal with that excess. So just give us a little bit of perspective about iron within within blood. So here we're looking at the distribution of iron within serum transferrin. As remember, transferrin is the primary binder of iron within the soluble part of blood. And so transferrin is the indicator of the amount of iron that we have in our body, and also is an indicator of if we're possibly in a diseased state. So just to give it a perspective, the protein itself is not 100% saturated with iron. Is actually a significant amount that is metal-free. So you have about 39% that is apo, so it doesn't have any metal bound to it. And then you have some amount that is actually saturated with the metal, and a significant amount that is only mono iron bound. So, but when I say mono iron, it could be either at the N lobe of the protein or the C lobe of the protein. So clearly, you don't have the saturation of protein. And so this is our way that, by knowing this general distribution, we have a sense of the amount of iron within the body. But when you do reach this excessive amount, these are some of the parameters you will see. You will have basically 100% iron-bound transferrin, and you will have an uptick in the amount of non-transferrin-bound iron. And so these labile extracellular species are the ones that can be quite toxic, again, as they can generate reactive oxygen species in a very uncontrolled, unregulated manner. All right, so what are the routes by which to deal with this excessive intake of iron? So, well, before we get there, how is it possible? Again, as I already mentioned to you, excessive intake in food is one mechanism. Another mechanism is actually by multiple blood transfusions. That's a notorious way in which iron can become quite high in the body. And if there are mutations of proteins that are responsible for the iron cellular uptake and transport. I've only given you sort of a simplified view of this idea of how iron gets into our cells, but there are a lot of proteins involved in this process, especially in the signaling for sensing how much iron is in our blood and sensing how much iron needs to be released. And so if there's mutations in any of these proteins in this pathway to really control the iron homeostasis, that can lead to this overload of iron. All right, so one of the sort of older classical ways of treating iron overload is actually by bleeding patients. So you probably have seen in movies or in stories this premise about bleeding patients because that can provide some cure, right? And so some of that is based on this idea of trying to remove an excess of metal. And so you also have chelation therapy that is quite effective. There are some there are some FDA-approved chelators for this purpose of treating iron overload. So in general, I just want to give this highlight the importance of iron. We at the very beginning of this particular lecture talked about where we see iron in different proteins and the types of functions that it can facilitate. And it turns out that the World Health Organization considers iron deficiency to be the #1 nutritional disorder worldwide. So this has been for years now. And it turns out that about 80% of diets are iron deficient. And so there are a number of iron-related diseases, such as sickle cell anemia. This is a genetic disease that results in deformed red blood cells. You also have thalassemia, which is a consequence of lower levels of hemoglobin due to the growth of abnormal forms of hemoglobin itself. And then you have, as I've mentioned, iron overload. For instance, hemochromatosis. 1 in 250 persons of Northern European descent have this. It's a genetic disease that results in too much iron being absorbed. And then you also have acquired iron overload. That's a consequence of multiple blood transfusions. Iron is also implicated in a variety of other diseases, heart and liver disease, diabetes. It's actually in the diabetes world. There appears to be some correlation as well with sort of iron transfer and so some disruption in the distribution of the actual metal there. Neurodegenerative diseases, we'll talk about some of this. Cancer, as iron is actually very important in terms of the proliferation of cancer cells and also restless leg syndrome. So yeah, as obviously iron is extremely important, and that's why our body has imposed mechanisms to try to control its homeostasis. All right, so any questions before we move on?
Yo tengo una, profesor.
I have one.
Okay,
Because
who was first? I think it was
a past class you mentioned about.
Yeah, go. Sorry, Javier. I think someone was before you.
Oh, sorry.
King first. Brenda, go ahead.
¿Qué relación guarda el hierro con la hemoglobina? Porque pues, si uno tiene una deficiencia de hierro, pues ahí te baja la hemoglobina, puedes tener anemia. ¿Cuál es el proceso, verdad, que relaciona la hemoglobina con el hierro?
Right, so that can be quite complex. So you probably saw some of the ramification of sickle cell anemia and how it can affect certain groups of people. So that's something that's observed quite a bit in African Americans. So it can be a number of things. Like you can have mutations in the proteins itself that's going to affect the red blood cells, and as a consequence of that, how oxygen can actually get bound to the metal center itself. So it may not necessarily be the metal itself getting trafficked to particular proteins, but again, some sort of mutation that can lead to, a bigger effect in terms of just the normal functionality of a particular iron-containing protein.
Okay. Gracias.
Have you had question?
Sure. Yeah. In one past class, you mentioned about bacteriostatics. So how does that play a role in this sweet spot of iron?
Right. So we're going to talk a little bit about this later in the course. This idea of nutritional immunity. So it turns out that our body has mechanisms to also respond to whenever there's a pathogenic organism in our body, so microbes. And so one of the ways, classical ways, when it comes to iron, is that we actually will suppress the iron that's freely available in our cells. So that becomes a little tricky. So you imagine if someone is assessing you medically and they see that your iron levels are low, it's very important to check if there's a possible bacterial infection, because one of the really detrimental things is to supplement that person with iron, because your body might actually be naturally responding to try to hold on to his iron to minimize what's available for bacteria to be able to be aggressive. And so your body has a way of dealing with that, but it's something that's supposed to be transient, not supposed to be for very long terms. But yeah, we will talk a little bit more about this idea of a nutritional immunity that our body has, because it's not just with iron dependent.
Thank you.
Yeah. Any other questions? Okay, so that's. So we're going to stick to very similar themes, but we're going to go a little bit back to fundamentals so that we can, get some of the terminology to be able to appreciate this idea of just regulating metal biological speciation. So again, this idea of controlling the balance of metals and what are mechanisms in place. So we're going to look at a few fundamental things that will be reviewed for several of you. And if it's not reviewed, it's fine. We're going to get that terminology down under our belt so that we can be able to understand some of these themes as we see later on in the course. So there were a few things that I didn't discuss initially about the coordination numbers, so of certain compounds. So it can be a little bit difficult to know exactly what coordination is favorable for certain metals and others, but we get a general sense based on the hard, soft, acid-base theory, what may be likely as far as interaction between metals and certain ligands. And so something that I like to emphasize is the idea that you can have a number of complexes that can range in terms of their coordination numbers and factors that can favor, for instance, coordination numbers that are on the lower end. So what I mean by that is coordination numbers that are typically lower than 4, and what might enable that. And then those interactions that are, let's say, that are on the higher end. So just to review some of this, typically when you do see coordinations with metals that are less than 4, this will be seen with metals that are soft interacting with soft ligands. And so examples you might see of copper interactions that are of a 3-coordinate nature. And so this is a typical thing that, even though copper can vary in terms of being oxidation state 1, which is soft, and oxidation 2, which is intermediate, but you get the sense of how you can predict that. Something that can also favor a lower coordination number are the presence of large bulky ligands. And so that, because of sterics, it can block the interaction of further ligation events. And then when you're thinking about small molecules and how you can try to regulate the coordination number, well, this is an important factor here. So here I'm going to be a little bit more specific about what type of small molecules. So this particular point has to do with whenever you have a coordination compound that is charged. And so if we are talking about a coordination compound that is cationic, to prevent something that has the interaction with that cationic species that's going to neutralize, because any compound that we isolate from solution will be neutral in charge. Well, to be able to do that, you have to have something that is a counter ion, in this case, a counter anion. Now, the thing about counter anions is that they supply a negative charge. So what would prevent that counter anion from serving as a potential ligand, right? A source of negative charge for the metal. And so something that would be able to do so would be a counter anion that is of a low basicity. Right. So essentially, if that charge is not something that has any ligation capability. So one example of that I'm going to give you is this particular anion, which is called, it actually has this funny abbreviation called BARF. It's a boron-based anionic species. Its function is to exclusively serve as a counter ion and not as something that can coordinate to the metal center itself. All right, so looking at the other end, how do we look at those conditions that can favor complexes that are of a higher coordination number? All right, so what I mean higher, we're talking about something that's greater than 6 when we're thinking about pretty high up there. All right, so here, in contrast to the lower coordination numbers, you're going to have the presence of things that are hard metals, hard ligands, typically. And so in order to satisfy this higher coordination number state, then you need to have the presence of small ligands, as now you're not going to have that as much of a steric restriction. And then this third point, again, very similar to what I was talking about for the low coordination number, when we're talking about compounds that are of a charged state. So in this case, we're talking about a compound that is going to be anionic, right? So its overall charge is negative. All right, so how do we interact that anion with a counter cation in order to, at the end, have this neutral charge? Remember, the counter cation, it will also be positively charged, just like the metal. But how does that counter cation not compete with the metal for ligation? And so in this case, that counter cation has to be of a low acidity, such that it doesn't really have this propensity to bind to the ligands that would bind to the metal, so that there wouldn't be any competition, or at least minimal competition, between the cation and the metal for interaction with ligands. All right, but with that being stated, most of what you see in terms of structure within the biological complexes are distorted geometries. And so a lot of what I've talked about in terms of the correlation between coordination number and geometry are very idealized conditions that probably are not going to be a good reflection of how you will see coordination within the biological context, especially given that more often you're going to see ligands that are non-homogeneous in terms of their the atoms that coordinate to the metal center. And if you're talking about proteins, you're talking about atoms that are coming from amino acids that are, so you're coming from different points of the actual protein itself. And coupled with that, you're going to have a lot of constraints in terms of where metal is going to coordinate and what types of bond lengths and angles are even going to be possible. All right, so just keep that in mind when you're thinking about coordination more in a biological context. And so what is an additional factor that can really affect all of this is actually the solvent in which you're looking at this. And so I'm going to tailor this to an aqueous solution, given that we're talking about things more in a biological context. And so, as I've mentioned before, water is an important ligand. It's often treated as a silent ligand. And what I mean by silent is that you tend to ignore that it actually is there, especially when you see literature reporting binding constants. You tend to ignore that perhaps it's also water molecules affiliated with the metal. All right, but we'll get to that point in a moment. All right, so whenever we think about metal ions in solution in water, they're actually not free, as in they're not just free cations floating around. They are typically going to be ligated by water molecules. If we're talking about iron 3+, iron 2+, even as many as 6 water molecules surrounding the metal center. So then when we start to talk about coordination of ligands to that metal center, then we have to think about this idea of the ligand binding competing with solvent binding. And so in this case, competing with water binding. So here I present to you a representation of this, where we have your metal aqua complex, right? And now you have this ligand that has an affinity for that metal. And so when that ligand binds, depending on how many ligands bind to the metal center, what you're going to have is the association of one or more aqua ligands from the metal center. And so here we have this depiction of a ligand that's bound to the metal, and you have the loss of what appears to be one water molecule. So probably that ligand, even though I'm being very general about it, may be monodentate. And so we have here then that equilibrium. And that equilibrium expression is basically representing the strength of that metal ligand interaction relative to the metal bound to the water. And so when I was mentioning to you that we often will treat the water as silent, and this is something you have traditionally have seen before, when you look at, let's say, acid-base events in water being the solvent, you don't really take that into consideration, and because it's so high in concentration in the background. So when we focus on this equilibrium, this process of the formation of the product, what we're looking at is basically the metal ligand concentration, the complex, that adduct, with respect to the concentration of the free metal, with respect to the concentration of the free ligand. And this, again, is at equilibrium, not keeping in mind that there probably are water molecules that are bound to the metal center itself. All right, so with that in mind, I want to focus on this idea of formation constants, and essentially the measure of the strength of ligand affinity for a metal, and overall a measure of the stability of the metal ligand complex. And so we can look at this from two perspectives, from a single step and cumulative step formation constants. All right, so what do I mean by a single step? Literally a one binding event. Whereas when I'm talking about cumulative steps, I mean you might have more than one ligand binding to a metal. And how do we represent that process? All right, let's start off very simple. We're going to look here at a single step metal ligand interaction. And so here we have our expression for one metal interacting with a ligand. And what we're not considering here in this representation is the denticity of the ligand. We're simply treating that we have this point of contact with the metal. And so we have one metal plus the ligand, giving you your metal ligand complex, your metal ligand adduct. And so the K value is your formation constant, or another way of saying that, your stability constant. And so that is our measure here. So as we're well aware, the higher this K value is, the higher the stability of the metal ligand compound. But now let's imagine that we have a second ligand that's going to coordinate to the metal center. So how do we represent this in terms of a single step process? So let's break this down. We have that first ligand bound to the metal. And so we will define that as the first step, which is K1 in this context. But then when we have a second ligand binding, well, now we have a complex that's already formed, ML, and that complex will interact with another ligand, L, okay, forming the compound M with the two ligands. So now this would be the second step, because that represents that second ligand now binding to the metal center. And so we represent this as the K2 equilibrium constant, which is the basically that representation of the amount of the metal with the two ligands relative to the original metal ligand compound and the concentration of the ligand itself, right? So that's how we will depict the second step, because again, we're looking at single ligand binding events. But we can also look at this from the perspective of the cumulative process, where both of those ligands will bind to the metal center. So how do we represent this idea of cumulative metal ligand interactions? So when we're talking about the simple metal interacting with one ligand itself, well, that in itself can be represented, as we already mentioned, as a K1 process, or we can use this terminology beta to represent that binding process. And it turns out that there's no other way that you can represent that one ligand and that one metal coming together. It's both a K1 or a beta 1 expression. They actually are equivalent. But once we start to consider the presence of two ligands interacting with the metal center in a cumulative fashion, now the beta expression actually deviates from the K expression. And so let's look at how that is. So when we have that one metal interacting with two ligands, right, in this cumulative process to form the product M with the two ligands bound to it, now we can use the beta expression to express this process, because now we're not breaking it down to individual binding events. We're depicting this as an overall process, so that beta 2 equals to the concentration of that product that we have formed, divided by the concentration of the metal, and then times the concentration of the ligand squared, right? So here now the beta expression is taking into consideration the cumulative process. Well, it turns out that beta 2 is actually the product of the multiplication of K1 times K2. So there are ways that you can, use the K expression to be able to derive the beta values. But you can see very clearly that beta is distinct from the K expressions when you're looking at higher order number of ligand metal interactions. So there's actually a general way that we can represent the cumulative process of metal ligand interaction. So here, what I'm showing you is this general way that we represent this, where we have a metal interacting with an X number of ligands, leading to the formation of this overall complex M coordinated to an X number of ligands. And so we use then this beta expression, beta X, which will equal to the final compound that's made divided by the concentration of the metal times the concentration of ligand raised to the X power. Okay, so that's our general way of representing cumulatively the metal ligand interactions. All right, so wanted to get that out of the way because we did see the use of beta in our previous lectures when we were talking about the siderophores binding to iron, and I had a list of beta values. So for those that were unfamiliar with beta values, it's important to review what beta values actually are. Okay, so now let's focus on those factors that are influencing formation constants. And so here, we're going to look at the different components, one of the major ones being the hard-soft acid-base theory. And so here, just to remind us, this premise of hard acids would prefer binding to hard bases, intermediate acids would prefer to bind intermediate bases, and soft acids would prefer to bind to soft bases. And so what we're seeing here is a table that reflects this preference. So we have two metals, iron 3 and mercury. So this is a representation of something that is hard. This is a representation of something that is soft. And so what we're looking at is the K1, the log K1, of the interaction of those metals with those individual anions. And so if you recall, when you focus on the periodic table, where the highlights are, as you go from the top to the bottom along that column, you're going to go from hard to soft. So what we're seeing is that range of hard all the way to over here being soft. And so as a consequence of that, we see the impact on the log K expression, where for the interaction between iron and fluoride, you have the highest log K1 expression, whereas for mercury, its interaction with the iodo ligand is going to be of the highest affinity. Right? And again, that's a reflection of this preference. Again, so I just wanted to remind you about the placements of the elements in the periodic table. Again, the breakdown in terms of what's considered an acid, what's considered a base, according to the Lewis acid-base theory, and then how we break it down into what's hard, what's intermediate, and what's soft. Okay, another really important factor for influencing formation constants is the chelate effect. And so we've already been talking about chelator, so we have an appreciation of what that means. But why does the chelate effect really improve the stability of binding metals? And so let's review this concept. It says, when coordinating through the same type of atom, a chelating ligand will outcompete a monodentate ligand for metal binding. Okay, so what that means is that a chelator version of a binding modality versus a monodentate version, the chelator will typically outcompete the monodentate version of that ligand. But there's going to be a few factors that are going to dictate this. So this chelate effect is favorable whenever you're forming a 5 or 6 member ring. And what do I mean by that? Well, what I mean by that is that you have, let's say, a chelator that when it coordinates to the metal will form this particular interaction. So what we're going to count is the metal, the coordinating ligands, and then whatever is along the the backbone of the ligand itself. So here we have what is a 5-membered ring that has been formed. Well, then you can also have this representation of a 6-member ring. We have here a total of 6 members, including the actual metal itself, right? So that's our 6-membered ring. So it's typically this arrangement of the chelator around the metal center that's going to be favorable for the chelate effect. So there are instances where you do form smaller rings, but typically the formation of smaller rings induce a lot of strain, so then that will not be as favorable. When you have ligands that might be able to coordinate and form larger rings, those tend to be quite unfavorable because it often will lead to ligand distortion. And so that's going to have quite a bit of penalty in terms of the formation of those complexes. All right, so now let's break this down into the components, the thermodynamic components of what's really underlying the chelate effect. And so here we're looking at the Gibbs free energy and the components of the Gibbs free energy. So delta G equals to delta H. Delta H is the enthalpy. So in this context, we're talking about either the contributions of bond breaking or bond formation. Subtracted by temperature multiplied by delta S being entropy, and entropy being that tendency towards disorder. So what we're going to focus on is, which of these, the enthalpy or the entropy, is really underlying the chelate effect. All right, so as our point of exploration of this, I'm going to focus on these two ligands that are amine based. So you have methylamine, and then you have there's diethylene amine. So basically we have the monodentate version, and then we have the chelator version. Because in terms of its point of contact, it's the same type of modality. Whereas here we have two the amines using their free pair of electrons to be able to coordinate to a metal center. All right, so let's look at this example right here, where we have this cadmium complexes that are coordination number 6, so octahedral complexes. So initially we have cadmium coordinated to 6 aqua ligands, and so we're seeing what happens when you have the methylamine coordinating to the cadmium center. So here we have cadmium. I'm just going to draw out the structure just to give us a perspective of the structure itself. All right, so we have these aqua ligands surrounding the metal center. And then for our product, then what happens is you have a loss of 4 of those aqua ligands, and they're actually replaced by this methylamine. Let's see. And so I'm actually going to put them as if they're going to be on this side here, excuse me. Didn't even do all that, but okay. So what you can see over here is the value of K. So technically speaking, this should actually be a beta expression because it's a cumulative process. All right, so we have a number that's 3.3 times 10 to the 6. So when we see this number, it's 10 to the 6 in terms of its order. It's favorable for the formation of this product here, right? So it's favorable for the binding of the 4 methylamines, and it leads to the release of the 4 water molecules. So what we're seeing here is an event where we have this original metal compound, right? So that the cadmium with the 6 aqua ligands, it has, we have the 4 methylamines interacting with it. It results in this complex here that's forming and the release of the 4 aqua ligands. So we have this balance of the total number of reactants and the total number of products that we have. So we have these 5 molecules over here, and then we have these 5 molecules at the end over here. Okay. All right, let's look at the next part. All right, so over here in this context. So now we have, again, the same cadmium complex, right, with the aqua ligands, but then we have its interaction with the ethylene diamine, which is this bidentate chelator. And as you can see, you require 2 of this ligand in order to be able to displace 4 of the actual aqua ligands. So let's draw the product that's formed here. Okay, so then we are forming this. All right, so what we're looking at now is the overall net effect of this. So let's draw our attention to this binding constant over here. So here now we have 4 times 10 to the 10. And so you can see in terms of the comparison with the monodentate version of this interaction, that this binding constant is 4 orders of magnitude higher, right? So that's quite significant. And so why is that the case? Given that we're essentially are binding the same type of, or we're essentially breaking the same type of bonds, and we're essentially forming the same type of bonds, right? So quick question to everyone. What type of bonds are we actually breaking? All right, so anyone, what bonds have been broken in this process?
El que está solo de H2, de agua.
What are they?
Ajá. the OH, this on.
So we're not breaking OH groups, but we are breaking the cadmium oxygen bonds. Those are what we're breaking. And so then what are the bonds that are formed?
What is format?
Uh-huh.
Filament?
So you can see right here, like for instance, here,
Yeah.
you're forming these
Oh.
cadmium nitrogen bonds. And so for both examples, we did the exact same thing. We broke cadmium oxygen bonds, the same number, and then we formed the same number of cadmium nitrogen bonds. So it's not what bonds are being broken or what bonds are being formed, right? That is actually is having the impact on the formation constant, on this beta value. So it turns out that when you look at it from the perspective of entropy, there is a difference. So if you count how many molecules you have here to how many molecules you have at the end, that is what's actually contributing to this drive. So on the left side, we actually have 3 molecules. And on the right side, we have the formation of 5 molecules. So we have a net increase in molecules that are being formed in solution of 2. And so it turns out that this process is actually being driven by entropy. It is not actually driven by enthalpy. Okay, so when we do this quick comparison, that's our net effect. So that's what we already looked at. And so it turns out that this process is entropy driven. Okay, it's very important to note that. When you're looking at monodentate ligand and a chelator, where the point of contact is the same coordinating atom. That's the key thing that we're focusing on here. All right, let's look at something that is related because of its chelation capability, but there's something different that's happening here. It's called the macrocyclic effect. All right, so here we're looking at this process of chelation, but it's something that's leading to a this cycle that's forming around the metal because of that binding process. And so if we draw our eyes to the figure that we have to the right, so where we're looking at these different copper 2 complexes that are formed where you have these nitrogen groups that are coordinating to the metal center. So these type of interactions make sense to us. We know that copper is something that's intermediate. Nitrogen is something that can be intermediate or hard depending on where it's positioned within actual ligand. All right, so these complexes basically make sense to us. The question is, why are these affinity constants different? So you have in this one to the left a log beta of 23.2. The one right next to it is 27.2, and you have this one here that is extremely high relative to the other ones, which is 38.1. So they are different, even though you're forming the same type of bonds, right? And so what's governing that? All right, so it turns out that this is actually going to be a process that's going to be enthalpy driven. And what really is enabling this complex here to have the highest stability is basically the planarity that's formed because of the positioning of those particular, that core that's interacting with that metal center is allowing for this concentration of the electron density there to be able to be well suited for coordinating that metal and basically really locking it in place, because that log beta value is quite high. And so you have that factor there that's really contributing to that formation of that particular complex. Additional things that we can consider when you look at that complex itself is the charge state. So when you look at this complex here, and you count up the overall charge, it turns out that you have something at the end. You have a neutral ligand interacting with a charged metal. So it leads to an overall cationic species. The same thing is true with the one to the right. You have neutral ligand interacting with a charged metal. The one at the bottom, you have this particular anionic species interacting with the cationic metal. So you have this boost of electrostatic attraction that can also be contributing to this, this more favorable complexation. Right, so you actually end up with a compound that is anionic. So there are a number of features that are occurring here, and that's overall was really enabling this macrocyclic effect. So a few things I'm just going to review here. So you have this highly versatile tetraaza macrocyclic ligand that's forming this very stable complex. You have the negative charge that's enabling this electrostatic attraction to the metal ion. And then you have the high rigidity because of that planarity as it coordinates to the metal center. So because of that high rigidity, you're getting that conjugated aromatic system that localized one, not localized, but you have the high electron density that's going to interact with the metal center. And so that's really what's helping to drive this macrocyclic effect. Okay, any questions about this? The Keeley effect or the macrocyclic effect? All right. All right, so now that we have some of these factors under our belt in terms of what's contributing to the formation of metal complexes, let's now look at this idea of speciation, and particularly as pH can really play a role in terms of having an impact on speciation. So oftentimes when we're looking at metal ligand complexes, we often will focus on sort of in a static format, like whether we're looking at it in a solid state format. But oftentimes when you take that same compound that has, can form something really stable in solid form and you put it into solution, now you can have a number of different species that might form, and that can be impacted by the interaction of solvent with the metal center. I've already discussed this idea of solvent playing a role with ligating metals, but also pH having an important contribution. And there is a contribution of the oxidation state of the metal itself, as we know, as that can dictate the hard softness of the metal. So when we're talking about this pH-dependent aqueous speciation, we're really talking about this distribution of metal species in water at different pH values under thermodynamic conditions. So for metal complexes, the focus would be on the different metal ligand species that can exist in water. All right, so to underline this, I want to really emphasize something about this process that certain metals can induce, and that is metal hydrolysis, because I have alluded to this a lot when I talked about iron, that iron, especially in its iron 3 form, is very hydrolysis prone. And because of that property, iron is very likely to become a precipitate in water. Well, let's look at how do we distinguish metals and this capability of hydrolysis? So what exactly is hydrolysis? So as the name implies, we're having a lysis of water, and that's actually being driven by the metal itself. So you have the metal compound, the metal aqua compound, and then the loss of a proton from the water molecule as induced by the metal itself. And so in this perspective, the way we're looking at this is the metal serving as a Bronsted-Lowry acid. So according to this, because it has the capability of releasing a proton, it's serving that function as a Bronsted-Lowry acid. And so here, what I'm listing to you is the relative acidity for different metals in terms of their capacity to serve, to induce metal hydrolysis, or their capacity as a Bronsted-Lowry acid. So when you look at this in terms of periodic trends, alkali metal cations, well, they are poor at this. They're not this type of acid at all. So when they interact with metals, you will not have this release of protons. But then when you move over to the second column of the periodic table, the alkaline earth metals, well, now these are slightly acidic. Then you have the transition metals, and here you have quite a bit of range. Those that are 2+ that have a weak acidity, those that are 3+ that have a moderate acidity, and those that are 4+ or even higher that have a very strong acidity. And because of that strong acidity, they tend to be very typically oxygenated. So you won't necessarily find them as their free form. They typically will be metal with some sort of oxide on it. So let me clear this up here. So for instance, vanadium is an example of a metal that's very metal hydrolysis prone. And so you'll often find vanadium 5 in its dioxyl vanadium form. So in that it exists as a vanadium oxide cation. So very important as part of this is that certain metals, when they interact with water or when they interact with ligands, have this property of being able to effectively lower the pKw of water, okay? Or they can effectively lower the pKa of ligands. So in other words, they can induce the dissociation of protons from water or the dissociation of protons from ligands at lower pH values than expected because of this capability. All right, so let's look at this with a little bit more context, the metal hydrolysis event. So here we're looking at an n number of aqua ligands on a metal, and then we have the release of a proton from one of the aqua ligands. And so now we have the formation of the corresponding hydroxide bound to the metal center. So often you will see in the literature a measure of the metal hydrolysis constant for different metals, and that's basically what this is representing, the loss of the proton from the aqua ligands coordinated to the metal center. Okay, so depending on how many protons are released is a measure of like whether it's a K1 event or K2 and so forth. All right, so I want to really emphasize this point of hydrolysis because it actually has an impact on how you can see certain metals in solution. And so I'm going to focus on a case study here on vanadium 4 and vanadium 5. And to really pinpoint like how even this one oxidation state difference can really lead to a difference in terms of the behavior, the speciation of this metal in solution. So here we're looking at vanadium 4 in the range of pH 3 to pH 11. And so this data, as calculated, I believe was for vanadium in the micromolar concentration range. And you can see that the vanadium is not going to remain constant throughout this broad pH range. So look at some of the features that we observed here. So at the low pH range, the pH of 3, we have one species here that is labeled as 100% vanadium 4. That means that you have 100% that species that exists in solution at a pH of 3. And so what is that species? It's this one right here, where you have the vanadium with an oxide. So you have what is called a vanadilyl ion. And then you have the corresponding 5 aqua ligands that are coordinated to the vanadium center. So you see you have this species that is cationic. As you start to go up to higher pH values, you see this particular species will decrease, and you start to get the formation of anionic species. Okay, and also in addition to being anionic, you start to see things that are of more than one vanadium nucleus. So you have this dinuclear species here that forms. So you get this transformation from low pH of cationic vanadium 4 species to high pH, where you now have this anionic vanadium 4 species that are formed. Overall, pretty simple speciation. But again, to highlight a few different things here. So from pH 3 to 5, you have a dominance of cationic species. Above pH of 5, you have the presence of more anionic species. This speciation profile consists of mononuclear. When I mean by mononuclear, I mean one vanadium, and also dinuclear, so 2 vanadium species. Now, also very important, although that's not illustrated in the figure, is that between a pH of 4 to pH of 10, the vanadium 4 has the capacity to be oxidized to vanadium 5. So it has that capacity, let's say if you open up to the air. But if you're looking at vanadium 4, let's say within the cellular environment, cells are quite reducing. So vanadium 4 is likely to exist as vanadium 4 throughout the whole pH range. So let's contrast this with vanadium 5. And you can see a profoundly different speciation for vanadium 5. It's much more complex, right? What you get is at the low pH range, so you're looking at something here, where initially it starts off as vanadium cationic species, so that vanadium oxide species, quite simple. But then as you increase in pH, you start to get these very higher order vanadium species, and many of these are anionic in nature. So this speciation is actually quite complex. So just to give you an assessment of this pH-dependent speciation, so at a pH of 1, you have that really simple cationic species, but really above that pH of 1, you have a dominance of anionic species. And in terms of the type of the nature of these species, you do have some mononuclear species, but the majority of what's depicted in this profile are multinuclear vanadium species. And so it turns out that in a reducing environment, vanadium-5 is very likely to get reduced to vanadium-4. So imagining putting vanadium-5 into an intracellular environment, that vanadium-5 is likely to get reduced to vanadium-4 if it's not protected by a ligand that has the capability of regulating that oxidation state. All right, so let's look at a few factors that really contribute to the metal ligand aqueous speciation, because what we were looking at over here are just examples of vanadium-4 and vanadium-5 in the presence of just water and how pH can dictate the speciation. But then when we start considering the presence of other ligands, so not the solvent itself, what are those factors that are going to have an impact on the overall speciation? So one of those is going to be the actual denticity of the ligand, so the chelation capability of the ligand itself. And then you also have the ability of the ligand to bind to the metal and whether or not that ligand has a protonation site. So for instance, there are a number of ligands that have atoms that are also sites of protonation that can actually result in dissociation of the proton. So you might have situation where, depending on the metal, the atom of the ligand will coordinate to the metal, so you have here, but also still retain the proton bound to it. But there are certain metals that, especially those that are hydrolysis prone, that when they coordinate to the ligand, you actually have the accompanying dissociation of the proton that was on the ligand, right? So it depends on the metal, and also, to an extent, this depends on the pH, whether or not the ligand, when it coordinates to the metal, will be able to hold on to that proton, or if it will actually release the proton itself. So as part of that, then the metal-induced hydrolysis, so the metal affinity for hydroxide ion, because that's a parallel thing, that can also contribute to the overall metal ligand aqueous speciation. So again, how the metal interacts with the solvent itself will have a factor. Another really important one are the concentrations of the metal and the ligand, because a lot of what drives the formation constants, so what drives the formation of the product is the amount of the metal and ligand that you have. And coupled with that is the ratio of how many ligands to metals are needed in order to satisfy the coordination of the metal. So that's going to be very important. In addition to the solubility product of all the species, so how soluble are these species in solution, are going to be things that are going to affect the overall speciation profile. So now that's just taking consideration ligand binding to metal in a water environment. But now let's try to get this a little bit more complex and start thinking about speciation in vivo. And now here, you're not just looking at one ligand interacting with a metal in water. Now you're thinking about water in addition to other things that are present that may have metal binding capability. So you can imagine that speciation is going to be far more complex in biological fluids. So keeping that in mind, there is a possibility that in terms of metal binding, you're going to have competition with low molecular weight species binding to metals. You might have competition with high molecular species like proteins binding to the metals. And another factor to keep in mind is how pH can also dictate the oxidation state of the metal. That's going to be very important. Again, as we saw with the vanadium case, vanadium-4, vanadium-5, you can have a favorable interaction with one oxidation and another depending on the environment in which it's located. All right, so I want to focus here back on vanadium-4 in terms of getting some idea about blood speciation of that particular vanadium oxidation state, but looking at specific vanadium-4 compounds. So here I'm looking at vanadium-4 compounds that have been reported to have anti-diabetic properties. So I'm not going to really go into any details of what are these anti-diabetic properties. That's something that we're going to discuss later in the course. But I just wanted to introduce this particular system to give us a perspective of speciation. So here I have a list of ligands that have the capability of forming vanadium-4 compounds that can exhibit anti-diabetic effect. And so let's look at these ligands and their coordination modality. So you have here picolinic acid. So here it's abbreviated as PIC. It has the bidentate capability. And then you have a derivative of that, the 6-methylpicolinate, also bidentate capability. You have the 2-hydroxypyridine N oxide that has that bidentate capability as well. And so a related ligand, which is a 2-mercaptopyridine N oxide, which has the bidentate capability, but there is that change in the OH to something that's softer to this phyl group here. We have maltol that has a bidentate capability here, and then the related 3-hydroxy 1,2-dimethyl-4-pyridione that also has that bidentate capability. All right, so just keeping that in mind, that all these have this bidentate nature, and so they can form very similar type of compounds. And so here I have a representation of the complexation they can form. So you have that 2 of these ligands will coordinate to the vanadium center, forming this square pyramidal structure. So I say square pyramidal, where you have the coordinating atoms forming this equatorial plane, and then you have the oxyl moiety that's in the axial position. And so you get these type of structures that you can isolate from solution, you can get crystal structure from them. So let's imagine we have these compounds and we're introducing them into the blood environment. So you're going to have a number of small molecules that have the capability of being metal ligands. So this is just a subset. There are many different ligands that do exist in the environment of blood. So you have citric acid that has the capability of binding metals through the hydroxyl group, through the carboxylic acid, the central carboxylic acid, or even at the end carboxylic acids. You have a lactic acid that has the capability of metal binding in a bidentate modality. Phosphate groups that also have the capability of metal binding. And so these are things that can complex metal speciation. All right. So with that in mind, here is data that was collected for a proposed speciation of taking these different vanadium compounds, so those 6 ligands that I showed you, and introducing them into an environment that contained citrate, lactic acid, the phosphate, in addition to 2 proteins, transferrin and albumin. And then what we're seeing is what happens when you have all of these different ligands, these metal ligand compounds, placing them into the solution that contains these small ligands in addition to those 2 proteins. And what you see in terms of this profile here are the different species that form. Okay, so we can try to break this down into what is actually occurring here. So it turns out, if you look at the data really closely, so you can see that plot there. The plot is of the percentage of vanadium of those different species. And so what are the species? You have vanadium bound to the protein transferrin. You have vanadium bound to the protein albumin, although we haven't actually talked about that protein just yet. We will talk about with more depth, but that's an important protein that can really have an impact on metal binding. You have vanadium bound to citrate, vanadium bound to lactate, and then you have a mixture of different species where you have vanadium bound to the original ligand that you supplied in solution, and also bound to the different anions that were placed in solution. So what does this overall plot show us? It shows us that all of these small molecule compounds undergo some degree of dissociation. So every single one of them actually did dissociate at least partially, okay? And then you get a mixture of the following. You get some amount of intact compound, but that seems to be a minority species in terms of all these compounds. If you look really closely at this plot, you have more the presence of what are known as heteroleptic compounds. What do I mean by heteroleptic? I mean, you have a mixture of ligands. So these compounds that you see over here, as labeled here, these are heteroleptic because they consist of the original ligand that you introduced into solution, in addition to the other ligands that were placed in solution as part of the sort of this fake serum solution that was prepared. And you also have exclusively vanadium bioligand species. So what do I mean by that? Basically, you don't have any of the original ligand of the compounds that you supplied in solution. That vanadium is either bound to the small molecules or protein bound, right? So this speciation became quite complex in terms of the mixture of different things that is actually formed in the solution environment. All right, so this is just the same plot that that I was just showing you, just to look at it a little bit more closely. All right, so what is the significance of this overall? So we have an appreciation of that compounds, when you introduce them into solvent, and especially when you introduce them into physiological solution, you're going to get a lot of complexity in terms of speciation. But this is actually really important, especially when you're thinking about the design of small compounds as therapeutics, because then you start to think about, well, how stable are they really when you put them in solution? So this is really important in terms of the gauging the pharmacokinetics, pharmacodynamics of compounds, but also thinking about their fate, their metabolic fate. So a number of things that we're really interested in knowing about biomolecules in terms of how do they regulate the particular metal in vivo. So a lot of this is going to be important in terms of understanding the solubility of these compounds. The oxidation state of the metal, again, that's going to be very important in terms of the overall stability in solution, and the potential role in cellular transport, biodistribution, and excretion. All that's very important. So will a compound that's designed for a particular therapeutic function, will it be able to reach its target? That's going to be important, because what you don't want is that, let's say you develop something for an oral delivery and it falls apart in the stomach. Well, then it's never going to reach its target if the idea is that it's going to get absorbed into the blood and you want it to reach a certain, let's say, a cell target. That may not be possible if it doesn't even survive the environment of the actual acidity of the stomach. So these are things to think about. And then can that particular, whatever the compound that you've developed and you've administered can actually be excreted from the body? Or how long does it actually stay in the body? So that's something that's really important to think about in terms of the speciation. So actually, I'm looking at the time here. I'm going to stop right here because I want to really highlight these points. We don't really have that many slides left here, but I just want to highlight these points with a little bit more emphasis in our next lecture. And I want to see if anyone has a question before we head out.
Oh,
Okay, no questions? All right, everyone. Well, have a great end of your week and a great weekend. Take care.
Have a great
bye.
Have a good day.
Hey, bye-bye.